Hi and welcome to SBP 4504. In this video, we will be explaining on the topic of genetic basis of drug metabolism that have been prepared by the HAVO. These two objectives will be the main focus in this topic. Before we go into details, what is meant by pharmacogenetic? Pharmacogenetic is a study of the role of genetic in drug response. And what is meant by genetic polymorphism? Genetic polymorphism can be defined as when a place in the DNA sequence where there is variation and the less common variant is present in at least 1% of the people. Here is our first example, which is G6PD. G6PD, or known as glucose-6-phosphate dehydrogenase, is used to maintain oxidation reduction in the cell. The absence of it will lead to deficiency and allow oxidative drug to oxidize and cause hemolysis. G6PD deficiency is a sex-linked recessive trait, and it is caused by one-point mutation where nucleotide conversion from cytosine to guanine occurred. The low activity of it resulting in reduced glutathione concentration in erythrocyte and subsequently caused hemolytic anemia following the ingestion of certain drug. G6PD deficiency is divided into two types of mutation, and it can be found commonly in African and Mediterranean lineage. Here is the list of drugs that cause hemolytic anemia for G6PD deficiency. In order to treat the cause, cessations of drug administrations is needed, and for severe cases, blood transfusion and corticosteroid administrations is required. Okay, let's move on to the next example, which is an acetyltransferase. An acetyltransferase is a phase 2 conjugating liver enzyme which catalyzes the N-acetylations and O-acetylations of amine carcinogens and heterocyclic amine. Na2 gene is responsible for fast acetylator phenotype. When 2 or 3 point mutations occur, it results to slow acetylator phenotype. When someone is having a slow acetylator phenotype and ingesting these drugs, it will increase the risk of getting peripheral nerve damage, idiopathic lupus erythematosus, and increase the risk of getting cancer. People with slow acetylator phenotype are most commonly found among Egyptian and Jewish, while East Asian and people at the north part are the least common.
The superfamily of cytochrome isoenzyme consists of more than 50 heme-containing proteins which catalyze the oxidative metabolism of many structurally diverse drugs and chemicals. It has an important role in phase 1 biotransformation. Cytochrome 2D6 isoenzyme metabolizes 25 to 30% of all clinically used medications, including dextromethorphan, B-blockers, antiarrhythmics, antidepressants, antipsychotics, morphine derivatives, and other drugs. Genetic polymorphisms cause reduction or complete loss of activity. Poor metabolizers possess 2 non-functional alleles, and it is an autosomal recessive trait. The frequency of poor metabolizers among ethnic groups. Poor metabolizers of cytochrome 2D6 may result in lack of analgesic effects from codeine, and standard antidepressant dosage becomes ineffective. Cytochrome 269 involved in the metabolism of warfarin, phenytoin, dobutamine, lutein, and NSAIDs. The variant alleles contain single nucleotide polymorphisms that result in single amino acid substitution. These allelic variants are associated with decreased enzyme activity. Patients with mutant allele has increased bleeding risk in standard warfarin dose, low blood sugar levels, slow metabolism of NSAIDs. Cytochrome 2C19 involved in metabolism of S-mephenethine, omeprazole, and other protein pump inhibitors, diazepam, propranolol, imipramine, and amitriptyline. The variant alleles in poor metabolizers arise from single base-pair substitution in exon 4 and 5. About 12 to 23% of Asian and 3 to 5% of Caucasians are poor metabolizers. Poor metabolizers of cytochrome 2C19 may result in cytochrome 3A subfamily play a significant role in the variability of oral bioavailability and metabolism of cytochrome 3A substrate. The variant alleles are the functional consequences of polymorphisms may result in increased or decreased activity of the isoenzyme. Thus, pharmacogenetics play an important role in predicting a patient's variability in drug activation and detoxification status. It may lead to personalization of dosing to improve therapeutic effect.